Well, let's go ahead and summarize what we've learned in this chapter. Gregor Mendel formulated a theory of inheritance based on the experiments with garden peas. The law of segregation states that genes have alternative forms, or alleles. In diploid organisms, the two alleles of a gene segregate during meiosis and gamete formation. The law of independent assortment states that the pair of alleles for a given gene segregates into gametes independently of the pair of alleles for any other gene. Inheritance patterns are often more complex than predicted by simple Mendelian genetics. This includes incomplete dominance, codominance, multiple alleles, pleiotropy, epistasis, and polygenetic inheritance. Environmental influences can affect expression of the genotype. An organism's overall phenotype reflects its overall genotype and unique environmental history. Analysis of family pedigrees can be used to deduce the possible genotypes of individuals and make predictions about future offspring. Many genetic disorders are inherited as simple recessive traits. Lethal dominance alleles are eliminated from the population if affected people die before reproducing. Many human diseases are multifactorial. That is, they have both genetic and environmental components and do not follow simple Mendelian inheritance patterns. Now, as a reminder of the learning objectives for this chapter, be able to define the following terms: true breeding, hybridization, monohybrid cross, P generation, F1 generation, and F2 generation. Distinguish between the following pairs of terms: dominant and recessive, heterozygous and homozygous, genotype and phenotype. Be able to use a Punnett square to predict the results of a cross and to state the phenotypic and genotypic ratios of the F2 generation. Explain how the phenotypic expression in heterozygotes differs with the complete dominance, incomplete dominance, and codominance expression. Define and give examples of pleiotropy and epistasis. Explain why lethal dominant genes are much rarer than lethal recessive genes, and explain how carrier recognition, fetal testing, and newborn screening can be used in genetic screening and counseling. Well, that's it for this chapter. Let me know if you have any questions.